So first of all, I'd like to thank Frank and the committee for inviting me here to speak. It's a real honor. I've never spent much time in Reno. I've always been curious to learn more about UNR and Desert Research Institute. So it's a pleasure to be here. Thanks also to those students who spent time with me, Allie, lunch and at breakfast. I've really enjoyed our discussions, and I'm looking forward to more this afternoon. Let's see. So what I'd like to talk to you about today is work that our DOE-funded National Lab Research Program has been doing looking at uranium and sediments. This is the old rifle uranium mill tailings remediation or processing site. And if there's time, I'll talk to you today about two major aspects of uranium biogeochemistry that we've been studying at the rifle site. The first is bioremediation, where we're trying to use engineering techniques to control the redox status of uranium in the subsurface. And the second is plume persistence. And in this case, it's the same thing. It's still biogeochemistry that's controlling the uranium, but it's nature that's doing it. And it's a much harder problem to study, but it's also extremely important. Okay, so I'm the lead PI for our National Lab Research Program, which is funded by Department of Energy Office of Biological and Environmental Research, and specifically within BER, the Subsurface Biogeochemical Research Program, which is alive and well. I'm very happy to report. And the mission of our research program is like this: is develop and inform biogeochemical process models. So how is it that we describe subsurface uranium behavior in biogeochemical models, as well as to provide regional scale insights for uranium behavior targeting the upper Colorado River Basin, and in so doing advanced DOE BER modeling competencies connecting the pore scale all the way up to the regional scale. So somewhat ambitious, with biogeochemistry being at the center of it. And I guess I should also say that the three major elements that we're trying to connect through this research are the behavior of natural organic matter, and also redox cycling of nitrogen, sulfur, and iron, and how those feed back into uranium behavior. All right, so it is a fairly good-sized team, and I just want to acknowledge the many team members that are part of this. What I will be showing you today is work, some of which I did myself, but much of which was done by our other team members. And just very briefly, the outline for what I'll talk about today is, first I'm going to talk about speciation and redox pathways during in situ biostimulation. So this is a bioremediation technique. And then if there's time, then I'd like to go on and talk about this other part where we're kind of using this as a platform to move on to studying this much harder problem of what is controlling plume persistence in the upper Colorado River Basin. All right, so just starting from the problem, what I'm showing here is a plot of all of the legacy management sites in the country. So there's multiple DOE organizations that administer contaminated sites in the U.S. The most important one, arguably, is the Department of Legacy Management. So all of these little blue dots and triangles right here are showing the sites which were former uranium ore processing sites during the 40s, 50s, 60s, and 70s. Uranium ore was produced there not only for civilian nuclear energy production, but also for weapons development. And many of these sites were closed after the 3 Mile Island incident in, I forget what it was, 1972, something like this, that caused the market price for uranium to drop so suddenly that all the major corporations that were running these uranium mines in this area out here, they just walked away from these mine or processing sites and left these large contaminated floodplains. So uranium is a very important inorganic contaminant at these sites. It's second only to tritium in importance. Treatment of these sites is nearly impossible because the uranium contamination is in plume form. It's in the subsurface. Typically, we're talking about millions to hundreds of millions of gallons of contaminated water at depth. And many of these plumes are persistent. And in fact, there's one other very important point, which is if you draw the geographical boundary for the upper Colorado River Basin on this map, what you can see immediately is that plume persistence at sites is in fact a regional problem, mostly residing within or immediately proximal to the upper Colorado River Basin. So one example of one of these impacted Umtrica sites is the Rifle, Colorado site. You saw a picture of that on the first page already. This is the Colorado River. Looking upstream here, you're looking up toward Aspen and Eagle. Highway 70 is just on the other side of the road. So the Rifle site it has 100 million liters of uranium contaminated water. The plume is persistent. That means that natural flushing will not attenuate this plume naturally within 100 years. This is more than a 100-year plume. And in fact, this site serves as a model for many other sites within the upper Colorado River Basin, which also have persistent plumes that will last for more than 100 years. Okay, so Old Rifle was a former uranium ore processing site. So this shows a picture around the time that it was shut down in 1957. And what you can see here on the east end of the site is the mill, and then you see these tailings sitting all around. There's a large pond impounded here. And just a few, and so this is what it looked like then, this is what it looks like now. A few key points: the uranium dissolved in the surface is concentrations around 1 micromolarity. It's mostly complex by carbonate and calcium. These materials here on the surface were removed in 1996, and the uranium concentration in the site is still about 8-fold higher than the EPA regulatory standard. Again, this is a common problem at sites in the upper Colorado River Basin, and it's more than a 100-year plume. Okay, so one of the original uses for the DOE field research site at the Old Rifle location was to study this process called bioremediation. And the basic idea behind bioremediation is that you use heterotrophic bacteria that have the ability to reduce uranium to change its solubility, so its redox status and its solubilities. So very simply, the idea is you have a contaminated portion of the aquifer. And let's see, what is this? Uranium-6 and you have this uranium plume in it. And the idea is that you inject a soluble exogenous electron donor like acetate or ethanol, something that stimulates bacterial growth. And what happens is you'll consume all the oxygen relatively quickly and develop initially metal-reducing conditions and subsequently sulfate-reducing conditions. So you essentially create a plume of acetate here in the subsurface. And right, the idea then is that within this plume, uranium is reduced from the relatively soluble and mobile 6+ oxidation state to uranium-4. Uranium-4 is much less soluble. So the idea is that by manipulating redox, using bacteria to manipulate redox, you can essentially suck the uranium out of solution, convert it to a solid form that'll be stable. And from the standpoint of the monitoring, you have a monitoring well at the site. You're measuring groundwater concentrations over time. The idea is that by applying this technology, the uranium concentration showing up in your monitoring wells would decrease below 0.044 microgram per liter. The site would be declared as remediated. You'd walk away. Well, I don't think they actually proposed ever using this technology to clean up the site. It's just that it was a really good place to do very deep scientific studies to understand the fundamental biogeochemistry of how this works. Okay, so I just want to point out that in addition to bacterial enzymes, inorganics are also very effective at reducing uranium. So this shows a series of redox couples between, for instance, oxygen and water. Here's manganese 2 and manganese 4, nitrate, nitrite. And the uranium redox couple would plot down here in this range of PE space. So this is just reduction of uranium-6 to uranium-4. And the key point I want to make is that this overlaps directly on top of the iron-2, iron-3 redox couple, which makes things really interesting and complicated. It turns out that reduced iron can reduce uranium, and oxidized iron can oxidize reduced uranium. So it can go both ways. It becomes quite complicated. Also, you can have reduction of sulfate to sulfide, and then sulfide can reduce uranium can also run this reduction reaction where you're taking uranium-6 and making it uranium-4. So that means that when you're stimulating the aquifer by dumping an acetate, bacteria may be making primarily iron-2 and sulfide, and it could be that the iron-2 and the sulfide is actually what's reducing the uranium and not actually the enzymes themselves. And this turns out is a very, very difficult problem to try and ferret out. One other thing I want to point out is that nitrite and nitrate are very strong oxidants that can oxidize uranium. So let's say you ran the experiment and you reduced uranium and everything was really happy. If later on you had nitrification and then denitrification running, the denitrification, nitrate itself as well as denitrification can actually lead to oxidation. And nitrate is a particularly interesting oxidant because it can diffuse, it doesn't react very strongly with sulfides, so it can diffuse into reduced sediments that oxygen cannot penetrate. All right, so the overarching questions that we looked at to try and understand uranium biogeochemistry were as follows. First of all, what is the speciation of uranium-4? What are the reductants that are acting on uranium? Is it enzymatic or are they abiotic? And this basically divides the world up practically into the two large camps. Most people who are studying uranium reduction were either very passionately convinced that it was a purely microbially driven process and enzymes are passing electrons directly to uranium and reducing it. And there's another large camp that pointed out very clearly and effectively that sulfides are a very powerful reductant and you don't necessarily need enzymes to do the job. And so this leads directly to the question, what sulfur and iron species are present, because these are incredibly important not only just to the uranium but to the other elemental cycles that are present for especially bacteria, because bacteria make their living by oxidizing or reducing iron and sulfur. Okay, so there have been pretty impressively, gosh, what's the word I'm thinking of? Sophisticated biogeochemical reactions, pathways, and networks that have been added into models now. This is a very simple conceptualization of what has been proposed and used to describe uranium reduction at the old rifle site. So the idea is that you have acetate that you're adding to the aquifer, and acetate then is consumed by iron-reducing bacteria or sulfate-reducing bacteria. And what happens is the bacteria directly pass electrons to uranium-6, reduce it to uranium-4, and uranium-4 precipitates as uraninite. Okay, so remember that. There will be a quiz on this later. So this is basically what the models assumed. And it turns out that this is it's very important to know this reaction product because the stability of that reaction product determines how effective ultimately the remediation technique will be. Ideally, you're going to make a very stable form of uranium-4, something that oxidizes very slowly. Furthermore, it was posited that the iron would be reduced abiotically to iron-2, which would react with sulfide. Sulfide is being produced over here by these sulfate-reducing bacteria. And you would precipitate iron sulfide. And another very interesting conclusion is that elemental sulfur would be produced as a result of this cycle. So there's the uraninite. There's the bacteria, so this is the biological aspect of the reaction sequence, and here are these other products. And so the question that we simply asked was, can we verify this model, but not just in the laboratory. We were very interested in trying to understand what was happening actually in the aquifer. And the reason why we wanted to know this was that many years of previous experimentation had already been done in the laboratory and by harvesting sediments and bringing them into the laboratory. And they had produced a series of conflicting answers. And we decided that if we really wanted to conclusively address this question, it was going to be necessary to do the experiment in situ. All right, so that required that we develop a new approach to observing these processes in the aquifer. And we did it in the following way. We basically took a bunch of these chromatography columns and packed them like you would to run an experiment in the laboratory. But what we did was we installed these in a well, and you can see how we did that in this diagram here. So the basic idea is that the chromatography column is sitting in the well, and the solution that is being eluted through it is just the groundwater. So we have a pump at the top. The pump is just applying a negative pressure to the system, and you're just pulling groundwater through this column as a function of time. We did add a little bit of additional uranium because we weren't sure if this experiment was going to work, and we wanted to just make sure that we would have enough reduction to be able to, and enough uranium accumulation. And the idea was that we would install it in the well gallery during an active field bioremediation test. So this wasn't just a laboratory experiment done in the field in a well. This is actually an experiment that was designed to sample all the evolving biogeochemistry that was occurring in the flow field during the acetate amendment experiment. So we had this particular set of samples located down gradient from an injection well. And the great advantage of doing this is it allows you to access the complexity that's present in that aquifer system that you absolutely cannot sample in the laboratory. And the reason for that is because these biogeochemical reactions are spatially resolved. So when you're injecting in an injection gallery in a well, you're creating a cloud of bacteria and also their products. So you're creating acetate. You have acetate. You're creating smaller molecular weight organic fatty acids. You're creating sulfide, and you're also creating. You're stimulating other types of communities. This is all occurring meters up gradient, and all of these different planktonic species are going right down gradient through the gallery. You can't simulate that in a laboratory column experiment because the up gradient end of your column is approximately 1 micron, right? You're just pumping reactants in and watching what happens when it blows throughout the other end. So you just can't do this in a laboratory. So, but by doing this in the field, we basically get direct access to the evolving aquifer conditions, the microbial communities, the reaction networks. We have the proper temperature, which affects the reaction rates and therefore the pathways that are selected, et cetera. And the basic idea was to do this experiment in the aquifer, harvest these things, and then forensically characterize the speciation of uranium and iron and sulfur using X-ray absorption spectroscopy, look at the pore scale distribution of the elements to get clues about the reaction mechanism, and to study the stability of the products to basically constrain future modeling efforts. And I just want to point out that, again, this is a large-scale effort involved collaborations with Rizlan Bernier-Latmani's group at University of EPFL in Lausanne, Dan Gimar's group at Washington University in St. Louis, Dan Alessi, who is now a faculty member at University of Alberta, Edmonton, whole slew of students and postdocs. So this wasn't just me. This was a large team effort. And what I just want to point out is when you start to stimulate bioreduction in the field, as well as in the laboratory, you actually get a fairly prolonged and complicated series of steps. So the first thing that happens is you get iron reduction. So this means the bacteria are passing electrons from acetate to ferric oxide minerals. And that's a fairly energetically favorable process. Eventually, however, for whatever reason, you begin to transition into sulfate reduction. And the reason why I say for whatever reason is because it's a little bit complicated exactly why the switch takes place. But this is about 3 weeks or so, 3 or 4 weeks. When you switch from iron-reducing conditions to sulfate-reducing conditions, you see a really, really big change in the geochemistry. So bear with me for just a minute. It'll get a little bit more exciting. So what I'm plotting here above are some of the key solutes. So sulfate is naturally abundant in groundwater at these Colorado River Basin sites, typically around 10 millimolar, but it can be up to 100 millimolar. So sulfate is present in the aquifer until you go into sulfate-reducing conditions. And then you can titrate all the sulfate out because it's forming sulfide. And that's this green curve here. You won't see any sulfide at all until you get into sulfate reduction and the sulfide comes up. Okay? So iron behaves quite differently than these two. In iron-reducing phase, what happens is you begin to produce quite an abundant amount of iron 2, and this is being produced because all the ferrihydrite and other types of iron 3 minerals are being reduced. You're producing this big plume of iron 2. And it can get up to concentrations approaching 200 micromolarity. But then what happens is once you start to produce this sulfide, the sulfide will react with this iron 2 and precipitate it out of solution completely. So what that really means is, again, during iron reduction, you have completely different biogeochemistry than you have during sulfate reduction. And in fact, you can separate the iron reducing stage into early and late. You can do the same thing with sulfate reduction. The point is that everything changes at the pore scale when you go between these biogeochemical regimes. The dominant bacteria change, the energetics that they're using to make their living changes, the local environment changes, the mineralogy changes. There are all these really major changes. And one of our initial hypotheses was, well, gee, with all these changes going on, we really expect that the speciation of uranium that's reduced during these conditions and the pathways by which it's reduced, it's pretty straightforward to predict that it should be very different than the processes in the species for uranium reduction under these conditions. So that was the basic zeroth order hypothesis. In order to test it, though, we needed to basically traverse all these different conditions. So we did that in 3 successive years of field experiments. The first one was in 2009. It was a feasibility experiment. We ran all the way to the very end of the process. After 100 days, we harvested the samples. Then we said, that worked. We were surprised. We were happy. So then we went back and we said, okay, great, we did that. Now let's really focus on this transition from late iron reduction to early sulfate reduction and try to test this hypothesis we had about different mechanisms working. And that worked. And so then we said, okay, well, let's go earlier and let's look to see if we can see any of the intermediates which we think probably exist during this time period. So the main point is we did the same experiment 3 times in a row, but we weren't doing the same experiment. So if you end up reviewing these papers, please don't ask me if we're just doing the same experiment over again. And the reason why it's not the same experiment is because different hypotheses, and in addition, it's a lot harder to do this experiment after just 4 days of reaction than it is after 100 days of reaction. All right, so let's see, what is this? So this is showing, this is the one, so this is just kind of coming right down to the punchline. So what I'm showing here are the results if we look at the speciation of uranium in these sediments after we conduct these experiments. Then we can use X-ray absorption spectroscopy to look at them. When we do that, we get peaks that look like this. So this is a so-called Fourier Transform. And this is telling us about the local molecular structure around uranium. And there's 2 primary model species that we use to look at these. The first one is uraninite, which I already mentioned. And the reason why uraninite is very important is, A, it's a very important ore mineral. B, it's predicted to be the dominant product in the modeling. And C, it's predicted to be the dominant product in the modeling because it is the most thermodynamically stable limiting phase. So if you collect the X-ray absorption spectra for this, the local environment should be dominated first by these oxygens in red. So if you look carefully, you'll see that every uranium here is surrounded by 8 oxygens, which are arranged on the corner of a cube. And in fact, you can see a nice large peak right here corresponding to those oxygens. And then going further out, you actually have 8 uranium atoms arranged around uranium at a much larger distance, 3.85 angstroms. And you get a peak corresponding to that here in the Fourier transform of the spectra. The other model that we have been using, this is based on many, many laboratory experiments where we're looking at bioreduction of uranium, is the so-called biopolymer-bound uranium-4. And the idea here is that uranium is coordinated to surface functional groups that are organic groups, probably containing phosphorus. And what you can see is that the spectrum for this looks really, really different than the spectrum for this. And the reason for that is that the local structures are really different. So bottom line is that we can use the spectroscopy as a fingerprint for the local molecular structure. And if you just scan through these spectra that we have from these different column conditions, what you can see immediately is they look a lot more like this than like this. So what this means is that sediment-reduced uranium-4 resembles this biopolymer complex uranium-4. And in fact, what really surprised us is that it does this across the entire range of conditions that we look at. So this is showing results from 2 successive years. No, this is just one successive. This is the Super 8 experiment. So you recall that we had first iron-reducing conditions, and then we had sulfate-reducing conditions, and our primary hypothesis would be that the speciation of uranium would be different under these 2 sets of conditions because the geochemistry and the microbiology is so different under these conditions. Okay, so what I'm plotting here in this line up here, this is showing, if you go back and look at the relative abundance of these 2 different species, the biopolymer uranium-4 and the uraninite, this is just plotting the relative percent of each of those species in terms of the biopolymer. So this is the biopolymer, or so-called non-crystalline fractional uranium-4. And what's really interesting is that it's not changing at all. In other words, the primary speciation of reduced uranium apparently doesn't know anything about all these massive changes in energetics and mineralogy and microbiology which are going on in the pore scale environment. So that was a very challenging thing for us to try to explain, and I'm not even sure I can convince people that we know why it's doing that yet. But what's really, so that's this point here. It's not, apparently not altered by geochemical conditions. What is really interesting is that the relative rate of uranium accumulation shoots up through the roof when you go into sulfate reduction. So these are the rates that we're getting under iron-reducing conditions, and these are the rates that we're getting under sulfate-reducing conditions. So that's kind of a conflicting set of results because on the one hand, the fact the speciation isn't changing doesn't really suggest that sulfide, for instance, is acting as a reductant, but the fact that the rates are going up so much under sulfate-reducing conditions suggests that sulfide really may be acting as a reductant because there's sulfide here and there's not sulfide here, and you can see the rates are much faster. All right, so if we look now at 2 years of experiments, so this is including, this is 2 full years of experiments, we see exactly the same thing. So this is our 2011 experiment, this is our 2008 experiment, and the main point that I'd like to make here is these are the rates that we get under iron reduction, and they're very, very similar even though it's 2 completely different years, different conditions. And when we go into sulfate-reducing conditions, we again get these much higher rates. So we basically verified that trend over a much larger range of time scales. All right, so the question then is how can we reconcile these observations? On the one hand, microorganisms appear to control uranium-4 speciation. Why do I say that? Well, it turns out that when you use sulfide to reduce uranium, the product you always get is uraninite. And we don't see very much uraninite at all. We're seeing biopolymer-bound uranium-4. If you use bacteria like Shewanella to reduce uranium, then you'll mostly get biopolymer-bound uranium-4. So this is why microorganisms really appear to be controlling uranium-4 speciation. However, as I just pointed out, the rate of accumulation apparently is quite sensitive to the presence of sulfide. So how do we try and address this? And the way we did it is by acknowledging that the one constant throughout the experiments is, in fact, the presence of microbes. So we're acknowledging that uranium appears to be bound by biopolymers, so biomass appears to be important throughout. All right, so if we bear with me for just a minute, I want to make another important observation, which is that if we use microscopy, we can get evidence for the reduction pathway. So what this is showing here is an individual mineral grain, and this is the iron elemental map over a 2 micron spot. So this is actually a 40 micron scale bar. So this is about 300 micron diameter grain, and it has a 10 to 20 micron thick iron coating around it. That's pretty common. And what we can do is we can go in at different points on this map and we can identify the different speciation of iron that we've got. And we can do that because by using X-ray absorption spectroscopy, we can distinguish between iron oxide, which looks like this, and iron sulfide, which looks like this. So if you do that, then you can get the iron speciation around this individual mineral grain. And this is a technique called X-ray microprobe analysis. We can do this at my laboratory using synchrotron-generated X-rays. And so what I'm plotting here is in red, this is showing iron oxide. And in green, it's showing iron sulfide. So this is actually pretty cool that you've got a grain with two really different redox conditions apparently existing next to one another. And the main point I want to make here is that if you look to see where the uranium is, you can do that by overlaying the uranium locations in blue. What you find out is that the uranium is mostly associated with the sulfide. It doesn't care about the iron oxide. Everyone knows uranium is, iron oxide is a strong sorbent for uranium-6, but in terms of the uranium-4, it's all ending up here with the sulfide, okay? So the uranium is spatially associated with the iron sulfide, and this suggests that iron sulfide is supplying electrons to reduce uranium-6 to 4. However, there's a problem, which I already mentioned in my last slide. I'll just remind you. In the laboratory, every time that you take uranium-6 and you react it with sulfide, you get uraninite, which we don't see. Okay, coming back to the same samples, if you use SEM to look at these coatings that are formed, you see these iron sulfide coatings here. This is where the uranium is accumulating, okay? And here, if you zoom in on this little V shape, you see all these little things here. And these little boxes are showing locations where we took EDS spectra, so we could actually quantify the relative abundances of iron, sulfur, and uranium. And so this shows, for all these different points, this is showing the correlation between sulfur and iron, and you see they're almost perfectly correlated. And that's because there's basically a single phase that's present, and it's iron sulfide, so they should be very correlated. So if uranium was being reduced and was physically associated with iron sulfide, then you would expect a strong correlation between uranium and iron, or uranium and sulfur, like this. But in fact, we don't observe any correlation at all. So here's uranium atomic percent versus iron atomic percent, uranium atomic percent versus sulfur atomic percent. And the point is that at the sub-micron scale, there is zero correlation spatially between uranium and iron. So this is a really interesting result, because it's telling you that even though at the scale of a whole grain, wherever you get sulfide, you also get uranium, what it's telling you is that within the scale of the individual coatings, they're not physically associated. In other words, they're both ending up in the same location because it's an environment where sulfate is being reduced and where uranium is being reduced simultaneously, but the sulfide is not apparently directly acting as a substrate to reduce uranium. So then we put together this new model, which goes like this. We believe that in any case we should have some enzymatic uranium-6 reduction that's going on. We know that because we see uranium reduction early in iron-reducing phase. And we also believe that we're growing quite a bit of biomass because we're pouring so much acetate into the aquifer. And so this is just a conceptualization of a biopolymer matrix on the surface of a mineral. This could be a cell here. These little feathery black things are supposed to be iron sulfides. If you zoom up on iron sulfides, if you do electron microscopy, they're pretty boring. They're these little platelets. And so our model basically goes like this. It turns out biomass is really, really, really good binding agent for uranium-6. That's why uranyl acetate is used as a stain for biological tissues in a SEM laboratory. So you can accumulate uranium-6 on this biomass, and then once it's stuck there, you can have it reduced. So the really interesting thing is, if you complex uranium-6, if it binds to these organic functional groups, and you can reduce it there, and then if the reduced form has a fairly stable structure, then there's no reason to precipitate uraninite. And not only that, but the electrons that are reducing the uranium, basically they can come from anywhere. They can come from enzymes, and they can also come from the iron sulfide, right? Because you can have electron shuttles, for instance, just direct dissolution, allowing iron and sulfide to come over here, make contact with the uranium and reduce it. So electron shuttles are required, but they're likely to be there. And this means the reductants can be abiotic or biological. The key point here is that you will get the same uranium product under any set of conditions, because uranium first binds to these organic ligands, and then it's reduced and it stays stuck to them. And that is one possible explanation for the otherwise somewhat perplexing behavior that we're seeing. So one interesting prediction that you make from this is that if you stopped adding acetate to the aquifer during bioreduction, you could continue to have uranium reduction because you've accumulated all this sulfide, and the sulfide can continue to act as reductant. And in fact, that is what's been observed at the old rifle site. One other point I just want to make is that the model that I showed you earlier predicts that sulfur should accumulate. You can also do X-ray mapping to see where the sulfur is. The sulfur is showing up in green. And the point here is that this thing is just glowing in elemental sulfur. So we can confirm those two parts of the model. All right, so this just summarizes that part of the talk. The first point is that uranium is being reduced. There probably is some enzymatic reduction. However, this is definitely, the model is assuming the wrong product. The product that needs to be incorporated into the model is this biomass-bound uranium-4. The other really interesting thing that's missing from the models is that you can also have this abiotically linked reduction pathway, which apparently has a massive effect on the rates. So there's no way a model is going to get the rates right unless that they have this proper reaction sequence in it. And we also observe iron sulfide as predicted and sulfur as predicted. Okay. So I just discussed these implications. There's an important modeling implication. The electron transfer pathways are likely to be flexible. This needs a lot more attention. Basically what this means is that you can't separate uranium reduction pathways into simple abiotic and biotic mechanisms. They're linked. They're intimately linked at the submicron scale. That has really interesting implications also for orogenesis pathways as well as isotope fractionation. All right, so I have just a few more minutes, and I think what I'm going to do is drag you all through what we're doing now. So the work that I just showed you, it's wrapped up. We've been wrapping it up over the last couple of years. And now what I'd like to do is zoom out quite a bit from the micron scale to the 100-kilometer scale. So this is the upper Colorado River Basin. And talk again about plume persistence. You remember at the beginning of the talk that I mentioned that uranium plumes tend to last for a very long time at this location. And what that's telling us is that there's a source of slow-release uranium to the aquifer, and we don't know squat about it. Okay. So the question that I'm going to pose here is, what can we learn about plume persistence in the upper Colorado River Basin from studies at Rifle? The answer is quite a bit. Okay. So it turns out, let's see, I was going to check to see how well you can see these pictures. They tend to project dark. I don't actually know how well you can see that. But you'll just have to trust me here if you can't see this well. One thing that was noted in the last 10 years of work at Rifle is that there are sediment lenses in the aquifer that are very organic rich. And so this is a good picture of them. They're black. They're black because they contain sulfides. There's a lot of recalcitrant detrital organic matter in them, like sticks. If this were brighter, you'd actually see lots of little root hairs sticking up here. And if you look at the oxidation state of uranium-4, of uranium in these sediments, it's present as uranium-4. And not only that, there's a lot of uranium-4 in these layers. This is just showing a vertical profile from the surface grade going all the way down to bedrock in this shallow, unconfined aquifer. In gray here, I'm plotting the organic carbon content. And you can see there's a lot of organic carbon here, almost 2.8%. Corresponding to the sedimentary material. And you can also see that the uranium picks up quite a bit. If you integrate this vertical profile, you find out that essentially all of the uranium in this vertical core is sitting in this thin organic rich layer. Okay. Also, 50% of the organic carbon is in there as well. So this is very important for overall carbon cycling in the floodplain. Okay, so see, I guess I'm going to skip over this for now. So it turns out then that these organic rich sediments are believed to be very, very important for biogeochemical cycling. not only of uranium, but also iron, nitrogen, and a bunch of other processes. And you can actually see that if you look at the transition. You see here it's light, here it's dark. This is the aquifer. This is going into this NRZ. NRZ means naturally reduced zone. The black coloration is from sulfides. It's telling you the sediments are reduced. So basically, wherever it's black, it's got sulfide and they're reduced. And what I'm showing over here are this so-called Zane spectra for sulfur. And up in this range here, you see this big peak indicating that there's sulfate. As you cross this transition into the reduced zone, you can see the sulfate peak goes away and these sulfide peaks appear. And one really, really interesting thing, so you have sulfate diffusing in, being reduced and accumulating as sulfide. And this is a biologically driven process. That's why I have a couple of bacteria here. This is telling you a couple of things. Most importantly, there's pretty good communication between these fine-grained, diffusion-limited, organic-rich zones and the overall aquifer. They're clearly exchanging mass, and they're also really biologically active. So they are biogeochemical hotspots. Okay, the other very interesting thing about them is their distribution. It turns out, rather than being thick layers, which we naively originally thought they were going to be, when we looked in detail, we discovered that they're these thin, discontinuous layers. And this is very important because if you're very thin, then it can improve the ability to communicate and exchange mass with the aquifer. Turns out the sediments are also very youthful. And another important thing is if you look at the speciation of uranium in here, you find it's not uraninite. You find, again, it's this biomass-bound uranium-4. And that's very important because it turns out biomass-bound uranium-4 is much less stable than uraninite. And as a result of that, this species is likely to be the most dominant control over uranium behavior in these sediments. So another very interesting thing is that because of all the productivity and all the reactions that can occur within these naturally reduced zones, they tend to be factories, biogeochemical factories. They're pumping reduced species out, like iron-2, uranium-6, and dissolved organic matter, and they're also acquiring sulfur and uranium. So we actually think these buried organic-rich layers are extremely important to the overall biogeochemistry, the overall they actually control, even though their abundance is relatively low in terms of volume, their impact is very large in terms of controlling the aquifer chemistry. This helps to explain why dissolved oxygen is so low, for instance, in these aquifers. All right, so our present model for these NRZs goes like this. You have these organic-rich layers. During the period of time when tailings piles were present and uranium was more abundant, you probably had uranium that was diffusing into these layers and being reduced from uranium-6 to uranium-4. There's some blue lettering here. You just can't see it. This says uranium-4. And now we think what's happening is this uranium-4 is being reoxidized and it's being released back out into the aquifer. And so this is very important because this could help explain why these plumes are so persistent. You just simply have built up this slow release, this relatively large concentration of slow release uranium, which is now subsequently coming out. So that's the basic conceptual model. Let's see, I can talk about depositional environment here, but we're running out of time. All right, just very briefly, the question is, where do these organic-rich sediments come from? So these pictures along the top here, these are from actual sediment cores at the rifle site. These pictures here are from modern-day stream banks, and the point that I'm making here is that they match. The textures match perfectly. So the idea is that these organic-rich chunks of material that are buried in the aquifer, they're simply dissected stream banks that have been buried as a result of alluviation and aggradational growth of these floodplains. So that seems pretty straightforward. So we can posit that these naturally reduced zones should be common throughout the entire upper Colorado River Basin. But that actually turns out to be very powerful because it leads directly to the hypothesis that uranium mobility and plume persistence region-wide is probably controlled by a relatively common small set of reactions involving this natural organic material. And in fact, if you go and look for evidence to support this conclusion, which we did last year, you do indeed find reports of organic rich sediments in sites throughout the upper Colorado River Basin. All right, so this then led to a series of experiments we proposed. I'm just going to jump over these quickly because we're running down on time here, and I want everyone to remember what I'm talking about, not remember what they were dreaming about when the clapping started and you woke back up, okay, which is what I always do. So if we come to this question here, so this is the big question: do NRZs regionally mediate uranium mobility in the upper CRB? If we establish this, then we can start to look in detail at the molecular scale reactions. And so to address this, our currently funded work includes as one module simply going out to these sites and doing very careful microbiology, geochemistry, isotope sampling, pore water sampling, etc. And we started to do that this fall. So this fall we went to the Grand Junction site and the Natarita site. And eventually we'll get down to Shiprock, Gunnison, and Riverton. It's off the map. Riverton is up here. And this just shows some pictures. Basically what we did was we went out to these remote locations and we set up a functioning field laboratory using duct tape and tent awnings and a few other things. And we just got our first set of results back the other day, and they're extremely encouraging. So what I'm plotting here is the fraction of the total uranium inventory in the core at different cores that we collected. And the main point here is that up to 70% of all the uranium in these cores is vertical cores going from the grade surface all the way down to bedrock. The majority of the uranium is present in these really thin organic rich layers, just like we saw at Rifle, just like we predicted. So this supports the idea that these NRZs can play a major role in uranium mobility region-wide. And so that's very exciting, I think, and very encouraging. So that means we'll move forward and we'll go to these other sites and look at the behavior. Another very interesting question that we're looking at is what really is the speciation of uranium? Knowledge of speciation in detail is essential to understanding and modeling its behavior and its transport properties. Non-crystalline uranium-4, as I already pointed out, it seems to be, so this is our representation of non-crystalline uranium-4 here. It's much more reactive than uraninite, therefore it's expected to control fate and transport, but we actually don't know what the heck it is. So this is another major effort that we have going on right now, is to try to understand if this uranium is bound to, for instance, lipopolysaccharide on bacterial cell surfaces, or to lipid bilayers that have been shed off of lysing bacteria and are now coating minerals, or something else other than that. That's a major research focus we have. Another major hypothesis regards oxidation. It goes like this. You can have nitrification going on. If you introduce a little bit of oxygen into these NRZs, then you can create nitrate. Nitrate can diffuse into these reduced zones, and it can be denitrified to nitrite, which is shown here. It's a bit difficult to see. Turns out nitrite is a really strong oxidant for uranium. So we think that denitrification is directly linked to uranium oxidation. And furthermore, that this process is driven by seasonal variations in the water table. This just shows the meltwater pulse that you get basin-wide in May and June. And the idea is that this alternately saturates and desaturates these organic rich layers. And that kind of change in saturation state strongly drives this nitrification cycling. Sorry, nitrification, denitrification, nitrogen cycling in general. All right, so just to wrap it all up, we do believe that natural organic matter is profoundly important to uranium behavior in the upper Colorado River Basin through direct complexation of uranium, influences on uranium speciation. We think binding to organic ligands modifies the redox couples for uranium. but we have much to learn about molecular pore scale mechanisms by which these biogeochemical cycles couple. And most importantly, it's worth continuing to investigate it. New discoveries of processes at molecular, pore, and aquifer scales have the very real possibility of leading to really significant advancements in, for instance, management of these uranium contaminated sites, much better predictions of behavior over time, and ultimately significant savings to taxpayers. And that's it. Finished on time, I think.